Hello, I'm Montana Smith, and in this video, I'll be explaining the difference between the two metadata fields, source material ID and sample link. In the sample metadata template, you will find source material ID here in column H and sample linkage in column I. In this video, you will learn the difference between the two and when and how to use them. When completing sample metadata, you may be submitting an experimental or an analytical sample. To identify which you have, think about the experimental process. When you run an experiment, you generate samples under some experimental context. For example, see here in pink, we have two samples that were incubated at different temperatures. These are your experimental or biosamples. This is your soil, culture, plant, water, or any other sample medium that has been generated with experimental intent. Analytical samples, seen here in yellow, are samples that have been extracted or prepped from your experimental samples for an instrument run and data analysis. For example, extracted DNA, digested peptides, or a prepared slide are analytical samples that are generated from the original soil, plant, or culture experimental sample. To describe how to properly use the source material ID and sample link columns, I'm going to walk you through this figure and an environmental sample example. Sample linkage is a one-to-many relationship to link multiple biosamples or experimental samples. For example, let's say you have a field sampling campaign in which you've harvested a plant and collected soil to take back to the lab to perform an incubation. For the plant samples, you've collected root and leaves from one plant. You should use sample link to link these two samples back to the parent plant. This is accomplished by using a globally unique identifier, or GUID, which you will hear more about towards the end of this presentation. See here on the sample metadata sheet for an example. The parent plant has been identified and linked to its child samples. Now for the soil you collected in the field, you've taken it back to the lab and performed a temperature and moisture manipulation. You've now changed the sample with experimental intent and generated a new sample. You can again use the sample link to connect the incubated sample to the field sample and the associated plants, if applicable. Similarly, if you have a culture sample and you've centrifuged the sample to separate the cells from the supernatant, you can link them using the same sample link field. There are many uses for this field, and if you have any questions or need clarification, do not hesitate to reach out to the metadata point of contact assigned to your project for assistance. Source material ID is a one-to-one relationship that captures the relationship between an experimental sample and its derived analytical samples. For example, if you perform an RNA extraction or a protein extraction and peptide digestion in your lab and are sending the analytical samples to EMSL, you would identify the experimental sample, the RNA, and the peptide sample on separate lines, as seen here. If necessary, you can indicate that the experimental sample is not being sent by indicating NA in the sample shipped amount, as I have shown here for sample 1. Here you will see that the globally unique identifier that I've assigned to the experimental sample is the source material ID for its corresponding analytical samples. This allows you to connect the experimental sample, or source material, to the two analytical samples that are of the same origin. Source material ID and sample link should provide a link to another sample and always be identified using a globally unique identifier, such as a UUID or an IGSN. For sample link, some prefix of the relationship you are defining between the samples should be provided. For example, parent, globally unique ID, or supernatant, globally unique ID. This ensures that samples are correctly associated with one another and the sample relationship can be made. GUIDs are not limited to UUIDs and IGSNs. Other options include ARC identifiers or RRIDs. For more details on GUIDs and identifier types, you can check out these publications. For more information about source material ID and sample link or any other questions, contact your project's metadata POC. And for more information and other video tutorials, visit the EMSL Learn website.